Dear students, hello and very warm welcome. Today I will be talking about the regulation of gene expression in eukaryotes. As you know, that gene expression is the process by which information present in the gene is transcribed to RNA and translated to functional gene product, that's the protein. Regulation of gene expression helps in control over structure and function of cells and is the basis for cellular morphogenesis, differentiation, and the adaptability and versatility of an organism. In eukaryotic cells, the ability to express biologically active proteins comes under regulation at several points, which include control at the level of chromatin and genome structure, control at the level of transcription initiation, control at the level of post-transcription initiation, translational control, and post-translational control. Now, let's discuss these levels of gene regulation one by one. First, let's know the control of gene regulation at the level of chromatin and genome structure. As you know, the physical structure of DNA, it exists compacted into chromatin, can affect the ability of transcriptional regulatory proteins termed transcription factors and RNA polymerases to find access to specific genes and to activate transcription from them. Coiling of DNA around a histone octamer in the nucleosome is now recognized as a cornerstone of transcriptional control. The degree of chromatin compaction essentially relies on histone modification and DNA methylation. Active chromatin regions usually contain high rate of acetylated histones and unmethylated DNA, whereas inactive regions are associated with non-acetylated histones and methylated DNA. Some gene expression pattern is independent of modifications in the DNA sequence, which includes epigenetic inheritance and gene amplification. Epigenetic inheritance refers to changes in the pattern of gene expression that are not due to changes in the nucleotide compaction of the genome and is essential to the normal development of eukaryotes. Some phenomena considered as epigenetic regulation include X chromosome inactivation and genomic imprinting. In chromosome inactivation, one X chromosome of the sexual chromosome pair XX is randomly inactivated at a very early stage of the embryogenesis. In genomic imprinting, DNA methylation prevents the binding of the transcriptional machinery, which leads to transcriptional silencing. A totally opposite process to chromosome inactivation and genomic imprinting is gene amplification that leads to the production of many copies of genes located in a spatial region of the chromosome. Each copy can be transcribed and translated, leading to an overproduction of the corresponding protein. This phenomenon occurs in normal cell growth in some species as well as in abnormal cell growth, such as in some cancerous conditions. Now, let's discuss the second and most important control level of gene expression in eukaryotes. That's the level of transcriptional initiation. The machinery required for generating so many patterns of gene transcription in eukaryotes has many components, including both regulatory proteins and cis-acting regulatory sequences. The first set of proteins comprises the large RNA polymerase II complex and general transcription factors. Eukaryotic nuclei contain three distinct types of RNA polymerases that differ in the types of RNAs they synthesize, which include RNase polymerase I, which is located in the nucleoli, synthesizes the precursors of all ribosomal RNAs except 5S rRNA. RNA polymerase II, which occurs in the nucleoplasm, synthesizes the mRNA precursors, and RNA polymerase III, which also occurs in the nucleoplasm, synthesizes the precursors of 5S rRNA, the tRNA, and a variety of other small nuclear and cytosolic RNAs. In addition to these proteins, a complex of at least 6 general transcription factors operate in eukaryotes. To initiate transcription, these proteins interact with DNA sequences called promoter proximal elements near the promoter of a gene. The second group of protein components consists of specific transcription factors that bind to cis-acting regulatory sequences in the DNA called enhancers or upstream activating sequences. These regulatory sequences may be located a considerable distance from gene promoters. Generally speaking, promoter proximal elements are bound by transcription factors that affect the expression of many genes, while enhancers are the targets of more specific transcription factors that control the regulation of smaller subunits of the gene. Often, an enhancer will act in only one or a few cell types in a multicellular eukaryote. For RNA polymerase II to transcribe DNA into RNA at a maximum rate, multiple cis-acting regulatory elements must play a part. The promoters, promoter proximal elements, and enhancers are all targets for binding by different trans-acting DNA binding proteins. The promoters of eukaryotic structural genes often contain a thymine adenine rich sequence called Tata box at a position of about 30 base pairs upstream from the first base pair transcribed RNA. The first transcription factor to bind to the promoter is the Tata binding protein, which, as its name indicates, binds to the Tata box and thereby helps identify the transcription start site. Tata binding protein is subsequently joined on the promoter by additional subunits to form about 770 kilodalton multi-subunit complex TF2D. One of these sites on the promoter is the CCAAT box, and often another is a GC-rich segment further upstream. Eukaryotic gene regulation mechanisms have been discovered through both biochemical and genetic approaches. The genetic approach has been advanced in particular by studies of the single-celled yeast Saccharomyces cerevisiae. For example, let's discuss the mechanism of gene regulation of galactose utilization in budding yeast, that is GAL system. In this system, to make use of extracellular galactose, yeast imports the sugar and converts it into a form of glucose that can be metabolized. Several genes, GAL1, GAL2, GAL7 and GAL10 in the yeast genome encodes enzymes that catalyze steps in the biochemical pathway that converts galactose into glucose. 3 additional genes, GAL3, GAL4 and GAL80 encode proteins that regulate the expression of the enzyme genes. The key regulator of GAL gene expression is the GAL4 protein, a sequence-specific DNA binding protein. GAL4 is perhaps the best studied transcriptional regulatory protein in eukaryotes. In the presence of galactose, the GAL1, GAL2, GAL7 and GAL10 genes are induced 1,000-fold or more. In GAL4 mutants, however, they remain silent. Each of these 4 genes has 2 or more GAL4 binding sites located 5' upstream of its promoter. The GAL4 protein has at least 2 domains, one for DNA binding and another for activating transcription. The modular organization of the Gal4 protein was demonstrated in a series of experiments to know whether a part of the protein is necessary for a particular function. When a form of the Gal4 protein lacking the activation domain is expressed in yeast, the binding sites of the upstream activating sequences are occupied, but no transcription is stimulated. The same is true when other regulatory proteins lacking activation domains, such as the bacterial repressor LAX-A, are expressed in cells bearing reporter genes with their respective binding sites. When a form of the Gal4 protein lacking the DNA binding domain is grafted to the LAX-A DNA binding domain, the hybrid protein now activates transcription from LAX-A binding sites. In Gal80 mutants, the Gal structural genes are active, even in the absence of galactose. This result suggests that the normal function of the Gal80 protein is to somehow inhibit Gal's gene expression. Conversely, in Gal3 mutants, the Gal structural genes are not active in the presence of galactose, suggesting that Gal3 normally promotes expression of the Gal gene. Extensive biochemical analyses have revealed that Gal80 protein binds to the Gal4 protein with high affinity and directly inhibits Gal4 activity. Specifically, Gal80 binds to a region within one of the Gal4 activation domains, blocking its ability to promote the transcription of target genes. The role of the Gal3 protein is to release Gal4 from its inhibition by Gal80 in the presence of galactose. Gal3 is a sensor and inducer. When Gal3 binds galactose and ATP, it undergoes an allosteric change that promotes binding to Gal80, which in turn causes Gal80 to release Gal4, which is then able to activate transcription of its target genes. Thus, Gal3, Gal80 and Gal4 are all part of a switch whose state is determined by the presence or absence of galactose. In a nutshell, the eukaryotic transcriptional machinery contains many proteins that are parts of various subcomplexes within the transcriptional apparatus that is assembled on gene promoters. One such subcomplex, transcriptional factor 2D, binds to the Tata box of eukaryotic promoters through the Tata binding protein. Gal4 binds to transcription binding protein at a site in its activation domain, and through this binding it recruits the TF2D complex and in turn RNA polymerase II to the promoter. The affinity of this interaction correlates well with Gal4's potency as an activator. Gal4 also interacts with the large mediator complex, which directly interacts with RNA polymerase II to recruit it to gene promoters. The mediator complex is an example of a co-activator, a term applied to a protein or protein complex that facilitates gene activation by a transcription factor, but that itself is neither part of the transcriptional machinery nor a DNA binding protein. The ability of activators to bind to upstream DNA sequences and to interact with proteins that bind directly or indirectly to promoters helps to explain how transcription can be stimulated from more distant regulatory sequences. Now, let's discuss another control level of gene expression in eukaryotes, that's the level of post-transcriptional initiation. The post-transcriptional initiation control includes the control of transcription elongation, the control of mRNA stability, and most importantly, the alternative splicing. After RNA polymerase II initiates RNA synthesis and successfully produces a short transcript, the transcription machinery undergoes a transition to the elongation mode. However, the transcription regulation rate is not always constant. In many genes, RNA polymerase II can pause during the elongation step. Pausing is influenced by the intracellular nucleotide triphosphate concentration, the presence of some transcription elongation factors, and the secondary structure of the growing transcript. Some pausing factors like DRB sensitivity include inducing factor and negative elongation factor appear to further stabilize RNA polymerase II in the paused form. Another mechanism is the control of mRNA stability. As you know, that the mRNA exported from the nucleus to the cytoplasm are translated until they are degraded. So, long-lived mRNAs give rise to more polypeptides than short-lived mRNAs. Thus, mRNA lifetime is another control point of gene expression. Actually, mRNA stability is one determinant of the efficiency of gene expression. However, the immediate products of transcription, the primary transcripts, are not necessarily functional. In order to acquire biological activity, many of them must be specifically altered by the exo- and endonucleolytic removal of polynucleotide segments, or by appending poly A tails to their 3' end, or by attaching a cap structure consisting of 7-methyl guanosine at their 5' end, and or by the modification of specific nucleotide residues. The longevity of mRNA is influenced by the poly A tail length that enhances the mRNA lifetime. The sequence of the 3' untranslated region preceding the poly A tail also affects mRNA stability. The concentration of some metabolites such as hormones can also influence mRNA longevity. However, decapping accelerates mRNA degradation. Now, let's discuss RNA splicing. RNA splicing is one of the RNA processing pathways leading to introns removal and exon splicing of a pre-mRNA. In some cases, introns removal and exon splicing can be done in different ways, leading to the formation of different mRNAs from an mRNA precursor. This phenomenon is called alternative splicing. There are 4 common modes of alternative splicing. Depending on the use of upstream or downstream promoter, the first or the second exon will be retained in the mature mRNA. Second, alternative use of the poly A sites determines the retention of the 3' exon or the previous exon. Third, intron retention. This mode of alternative splicing can have striking effect if retained introns contain a stop codon. This will give rise to a truncated protein when the mRNA is translated. Fourth, alternative splicing of internal exons. This gives rise to different mRNAs which encode different polypeptides with similar or totally different functions. Splicing regulatory proteins can usually act in a tissue-dependent manner. For example, in alpha-tropomyosin genes. In this case, tissue-specific factors determine the splicing pattern by choosing the 5' splice site and 3' splice sites to be used. Now, let's discuss another control level of gene expression in eukaryotes, that is the level of translation. Since many mRNAs have multiple methionine codons, the ability of ribosomes to recognize and initiate synthesis from the correct AUG codon can affect the expression of a gene product. Another translational control concerns proteins involved. As an example, let us discuss the iron metabolism. For example, ferritin has a function in iron storage, whereas transferrin receptor is responsible for iron import into the cell. When cytosolic concentration of iron increases, the synthesis of ferritin increases in order to bind the extra iron, whereas the synthesis of transferrin receptors decreases in order to import less iron across the plasma membrane. Both translational processes are regulated by one regulatory protein, aconitase, which can bind iron. Aconitase can recognize and binds to a stem loop structure at the 5' untranslated region of ferritin mRNA. This binding blocks translational initiation of ferritin mRNA. Aconitase can also bind to similar stem loop structure located at the 3' untranslated region of transferring receptor mRNA. This binding enhances mRNA stability. Aconitase dissociates from the mRNA when it binds iron. Thus, when iron concentration rises, translation of ferritin mRNA is initiated, whereas transferring receptor mRNAs are rapidly degraded. Post-translational control of gene expression can be defined as a regulation process in which protein structure and thus function are modified after translation. These modifications involve chemical modifications of amino acids, alteration of the order of amino acids in the polypeptide backbone, and others. The most common chemical modifications of amino acids include phosphorylation, glycosylation, and ubiquitination. The polypeptide backbone itself can be altered. The most common examples concern some large protein precursors which are cleaved after translation to give rise to active forms, for example, insulin, trypsin, etc. RNA editing is a process in which the RNA sequence is modified. The two editing mechanisms include site-specific deamination, in which a cysteine residue within the RNA is changed into uridine by deamination. For example, mammalian apolipoprotein B gene transcription with and without RNA editing in this case gives rise to two tissue-specific proteins. Guide RNA directed insertion of uridine, in which many U's or uridines are inserted into the transcripts as found in the mitochondrial mRNA of trypanosomes. These insertions completely shift the reading frame and give rise to totally different proteins. The expression of some genes are regulated by RNA rather than by proteins. An example is the attenuation regulation of E. coli tryptosine genes. Riboswitches, which are regulatory RNA elements, work in a similar way through their alternative secondary structures. RNAs can regulate gene expression in many ways. Short RNAs can repress gene expression by a precursor called RNA interference. RNA interference can intervene at the transcriptional or translational level and mRNA stability. RNA interference has diverse biological functions. These are short RNAs of about 23 base pair long and the products of long double-stranded RNAs which are cleaved by the nucleus dicer. These short interference RNAs direct a nucleus complex called RN-induced silencing complex to the repression of gene expression by three ways. First, destroy mRNA having complementary sequence with the short interference RNA. Second, inhibit the translation of the complementary mRNAs. Third, induce chromatin modifications within the promoter of these homologous genes leading to their silencing. MicroRNA is another class of regulatory RNAs. There are about 21 to 22 nucleotides long sequences and are produced from the digestion of larger transcripts by dicer. MicroRNAs can also destroy or inhibit the translation of target mRNAs which have homology of the microRNAs. There are hundreds of genes encoding these microRNAs in some eukaryotes. Most of them are involved in developmental regulation. Thus, we came to know that due to organizational characteristics of eukaryotic cell and organism and the spatial and temporal separation of transcription and translation, the regulation of gene expression in eukaryotes is much more complex and can be exerted at more than one level. With this, I conclude. Thank you.